Welcome to the PubMed Daily Review for March 21st. Today we scanned 4,438 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. NIR-II nanomedicine-engineered CRNK cells for precision navigation and potentiating lung cancer immunotherapy by remodeling tumor microenvironment by Yanning Dai and Qi Zhao from Cancer Center, Institute of Translational Medicine, Faculty of Health Sciences, 59193 University of Macau, Taipa, Macau, CR 999078, China. Let's break down its key contributions. Chimeric antigen receptor NK, or CRNK, cell therapy has been a game-changer for certain blood cancers. However, its success in solid tumors, particularly lung cancer, has been limited. The main hurdles are getting enough of these engineered cells into the tumor and overcoming the hostile, immunosuppressive environment within the tumor itself. This study explores a novel approach to tackle these challenges. To address the limitations of CRNK therapy in lung cancer, the researchers developed a sophisticated biohybrid system. They engineered CRNK cells and attached them to nanomedicine particles loaded with a near-infrared-2 dye and a SMAD3 inhibitor. This CKPSI construct allows for real-time tracking via NIR-II imaging, uses mild heat from NIR-II excitation to break down physical barriers in the tumor, and releases the inhibitor to remodel the tumor microenvironment. The goal was to improve CRNK cell delivery and function. The results were highly encouraging. The nano-engineered CRNK cells showed significantly enhanced infiltration into lung tumors compared to controls. This improved delivery, combined with the photothermal and SMAD3 inhibitory effects, led to a marked reduction in tumor growth. The treatment effectively remodeled the tumor microenvironment by reducing extracellular matrix deposition and boosting the immune activation of the CRNK cells, demonstrating a powerful synergistic effect. This study presents a significant advancement in CRNK cell therapy for solid tumors. By integrating imaging, thermal ablation, and microenvironment remodeling, this nanomedicine-engineered approach offers a powerful new paradigm. While these findings are exciting, further research is needed to translate this innovative platform into effective clinical treatments for lung cancer patients and potentially other solid tumors. Up next, our second paper out of 5. APO3 astrocytes can rescue lipid abnormalities and dystrophic neurites of APO for human neurons by Delara, O'Halim, and Oli Saxon. Let's break down its key contributions. Neurodegenerative diseases, like Alzheimer's, are increasingly linked to problems with lipids, not just protein buildup. Astrocytes play a crucial role by producing apolipoprotein E, or APO, which helps manage lipids in the brain. The APO4 gene variant is a major risk factor for Alzheimer's, and it's associated with issues in how the brain handles fats. This study investigates if a different APO variant, APO3, expressed in astrocytes, can protect neurons from these lipid-related problems. To tackle this, researchers created a sophisticated lab model using human neurons and astrocytes derived from stem cells, engineered to express either APO3, APO4, or no APO. They then mimicked a cellular lipid stress condition by blocking a key cholesterol transport protein. Using advanced live cell imaging and other techniques, they observed how the different astrocyte types affected the neurons under this stress, looking specifically at lipid buildup and the health of neuronal extensions called neurites. The results were quite striking. When the cells were stressed with lipid accumulation, the neurons co-cultured with APO3 astrocytes showed significantly less neurite damage and lipid buildup compared to those with APO for astrocytes. The APO3 astrocytes also produced larger, seemingly healthier lipoprotein particles in the culture media. Essentially, APO3 astrocytes demonstrated a clear protective effect, rescuing the neurons from the harmful effects of lipid imbalance. This study provides compelling evidence that the APO isoform expressed by astrocytes has a direct impact on neuronal health, particularly in the context of lipid metabolism. It suggests that enhancing the protective functions of APO3 astrocytes, or counteracting the deficits of APO for astrocytes, could be a promising strategy for treating or preventing neurodegenerative diseases. While these are early findings in a lab model, they open exciting doors for future therapeutic development and biomarker discovery. Up next, our third paper out of 5, Efficacy and Safety of Psilocybin in Treatment-Resistant Major Depression. The episode Randomized Clinical Trial by Leah J. Mertens and Gerhard Grunder from Department of Molecular Neuroimaging, Central Institute of Mental Health, Medical Faculty Mannheim, Heidelberg University, Mannheim, Germany. Let's break down its key contributions. Major depression is a significant challenge, and for many, existing treatments don't provide full relief. Psilocybin, a compound found in certain mushrooms, has shown potential in early studies. This trial was designed to rigorously test if a specific dose of psilocybin, combined with psychotherapy, could offer a new option for those with treatment-resistant depression. This was a Phase 2b, randomized, triple-blind, active placebo-controlled trial conducted at 2 centers. We enrolled 144 participants diagnosed with treatment-resistant depression who had stopped their antidepressant medications. They received either 25 milligrams of psilocybin, 5 milligrams of psilocybin, or a placebo, given twice, 6 weeks apart, alongside psychotherapy. The main goal was to see if the higher dose of psilocybin led to a significant reduction in depressive symptoms by week 6. The results showed that 17% of participants receiving 25 milligrams of psilocybin responded to treatment, compared to 10.6% in the placebo group. However, this difference did not reach statistical significance, with a p-value of 0.19. Secondary analyses did suggest a clinically meaningful effect, and the treatment was mostly well tolerated. Some safety concerns were noted, including increased reports of suicidal ideation and 2 serious adverse reactions. In conclusion, this trial suggests that psilocybin, at a 25 milligrams dose combined with psychotherapy, may offer a clinically meaningful benefit for individuals with treatment-resistant depression, even though the primary statistical goal wasn't met. While the treatment appears generally safe, vigilance for adverse events like suicidal ideation is crucial. These findings, while inconclusive, add to the growing body of evidence supporting psilocybin's potential and highlight the need for further investigation. Up next, our 4th paper out of 5, CRISPR-assisted probiotic and in situ engineering of gut microbiota, a prospect to modification of metabolic disorders by Raham Ramadi and Iraj Alaporford from Research Committee, Sharkard University of Medical Sciences, Sharkard, Iran. Let's break down its key contributions. The trillions of microbes in our gut, known as the gut microbiota, play a vital role in our health. They influence everything from digestion to immunity and even brain function. However, when this microbial community becomes unbalanced, a state called dysbiosis, it's linked to the development of serious conditions like metabolic disorders. Recent advancements in gene editing technology, particularly CRISPR, offer exciting new possibilities for precisely manipulating these microbes. This review explores how the powerful CRISPR gene editing system can be applied to the gut microbiota. The core idea is to use CRISPR/Cas technology to precisely target and modify the genes of bacteria living in our gut. This technique allows for specific genetic alterations, opening doors to understanding and potentially treating conditions linked to microbial imbalance. The results discussed in this review highlight CRISPR's remarkable potential. Its precision in gene editing is exceptionally high, allowing for targeted modifications within the complex gut environment. This technology is broadly applicable to various metabolic disorders, offering a novel therapeutic avenue. Furthermore, CRISPR is noted for its simplicity and effectiveness, making it a promising tool for future research and clinical applications. In conclusion, this review underscores the significant promise of using CRISPR to engineer the gut microbiota for treating metabolic disorders. While challenges remain in clinical translation, such as optimizing delivery and ensuring safety, the potential for developing personalized therapies with CRISPR-edited probiotics is immense. This technology could revolutionize how we approach gut health and metabolic diseases. Up next, our 5th paper out of 5, lenacapavir prevents production of infectious HIV, won by abrogating immature virus assembly by Clifton L. Rakana and Robert A. Dick. Let's break down its key contributions. Lenacapavir, or LEN, is a promising drug that targets HIV-1. We know it interferes with the virus's life cycle at various points. While its impact on the early stages of infection is somewhat understood, how it affects the virus's ability to assemble and produce new infectious particles has been a mystery. This study dives deep into that late-stage disruption. To investigate LEN's effect on virus assembly, the researchers used a combination of cutting-edge techniques. They employed cryo-electron microscopy to visualize the virus particles at an atomic level, alongside in vitro assembly experiments and assays within infected cells. They specifically used a modified HIV-1 clone that traps the virus in an immature state to better understand LIN's impact on capsid formation. The study revealed that lenacapavir treatment dramatically altered the virus assembly process. In a protease-defective virus, which normally remains immature, LIN induced a premature lattice. This unnatural structure resembled a mature capsid, but critically lacked inositol hexakis phosphate, or IP6, a molecule essential for proper viral assembly. Ultimately, this disruption effectively abrogated the production of infectious HIV-1. So, what does this mean for treating HIV? Lenacapavir doesn't just block the virus. It fundamentally breaks its ability to assemble properly, creating a non-infectious particle. This detailed look at the mechanism, particularly the role of IP6, is crucial. It deepens our understanding of how LIN works and reinforces its potential as a powerful, long-acting treatment option for HIV.